that's the point. So we move to the RNA. The RNA is a single-stranded molecule. So that means that you don't have it pairing with another. You don't have one strand pairing with another strand. So because it is a single-stranded molecule, it is a polypeptide because you have several repeating units of the nucleotides combined to form that strand. It also carries genetic information in certain organisms, for example, the RNA viruses. Still, the three components still remain. You have a sugar, a phosphate group, and then the nitrogenous bases. Okay, the difference is that with the nitrogenous bases, you don't have thiamine, but instead you have uracil. So uracil replaces uracil in the RNA. Okay, the fact that it is a straight, it is the fact that it is single-stranded does not mean that it can't fold to form complex shapes. But of course, when it does that folding, it has some hydrogen bondings happening, and that also contributes to its stability to some extent. Okay, so the ability, the RNA has a unique ability to repair itself, and as we shall see that if you have errors to do with how the nitrogenous bases are arranged, you will have errors in the nature of proteins that will be synthesized. So you don't want that to happen. But the RNA has the ability to repair itself. Okay, and that also happens in DNA. The DNA can repair itself. That is, if there are errors or misplaced nitrogenous bases. Some of them can be bases that don't really represent something, so they can be removed. So, like I said, the nitrogenous bases have to follow particular pairing rules. That in the DNA you have adenine with thiamine and cytosine with guanine, and this pairing happens because of hydrogen bonds that are formed, depicting the hydrogen bonds there. In the RNA, you have adenine bonding or pairing with uracil and cytosine with guanine. Okay, so this base pairing is very important because it ensures integrity and proper transfer of genetic information. Okay, so you have one nucleotide being connected to another via phosphodiester bonds, and this is a condensation reaction where you have water being given out. Okay, and this follows the particular direction. That's why we talked about the 5' and then the 3'. And usually for the growth of the polynucleotide chain, it is from 5' to 3'. The chains can have different numbers of the nucleotides. A shorter polynucleotide, which can have about 20 nucleotides. Then we can term that as oligonucleotide. For a polynucleotide that is actually longer, we say it is a polynucleotide where you have a long sequence of these nucleotides, and this happens in DNA and most RNA. Okay, so what's the role of the RNA in protein synthesis? The RNA is the one that copies the information from the DNA and then carries it out of the nucleus for protein synthesis to basically happen. And that copying is what we call transcription. But of course, there are different types of RNA molecules that do different roles. So the messenger RNA is the one that carries that information from the DNA and takes it to the ribosomes where protein synthesis will happen. And then the other type of RNA is the transfer RNA, where you will have amino acids picked from the cytoplasm being assembled such that you can have a particular protein being synthesized. And then the ribosomal RNA is part of the ribosome where you actually have protein synthesis happening. So far we are saying that the DNA is double-stranded, whereas the RNA is single-stranded. And in terms of nitrogenous bases, DNA has thiamine, whereas RNA has uracil, replacing it. But the other nitrogenous bases remain present in both RNA and DNA. The sugar that we use, that is used in formation of these nucleic acids, is that for DNA you have deoxyribose sugar, and then with RNA you have ribose sugar. Okay, it's the DNA that stores the genetic information, and the RNA largely transmits this information from the DNA to the ribosomes for synthesis of particular proteins. And RNA can be found in the nucleus and also the cytoplasm and also the ribosomes, but the DNA is largely in the nucleus, though you can have organelles that are outside the nucleus but still have DNA. So that is what we term as extranuclear DNA.